at 40. Okay, welcome everyone. So this is Jason, Associate Professor at UB Department. So today, for this session, we have three presentations, four speakers, because the first presentation will be delivered by two speakers. So let me first introduce the first two speakers. Their talk will be on the Internet of Nano Things, bridging the biological and the digital worlds. The first speaker will be Dr. Josep Jonet. So he was a professor at UB department, also a very good friend of all of us. So he is currently an associate professor in the Department of Electrical and Computer Engineering at Northeastern University, the director of the Ultra Broadband Nano Networking Laboratory, and a member of the Institute for the Wireless Internet of Things and the Smart Center. His current research interests are in terahertz band communication networks, nanophotonic wireless communications, wireless Nano-Bio sensing networks, and the Internet of Nano Things. In this area, he has co-authored more than 160 peer-reviewed scientific publications, including one book and 3 US patents. His work has received more than 8,600 citations as of November 2020. Since July 2016, he is the editor-in-chief of the Elsewhere's Nano Communication Network Journal. He is serving as the leading PI on multiple grants from U.S. federal agents, including the National Science Foundation, the Air Force Office of Scientific Research, and the Air Force Research Laboratory. He received the National Science Foundation Career Award in 2019, and he is the recipient of several other awards from IEEE, ACM, and UB. So this is Dr. Jonet is our first speaker, and so he will present give the presentation again together with Dr. Sasi Balasubramani. So he received a BE degree from in electrical and electronic engineering, and the PhD degree from the University of Queensland in 1998 and 2005 respectively, and the ME degree in computer and communication engineering from the Queen's University of Technology in 1999. He is currently the director of research with the Telecommunication Software and System Group, Waterford Institute of Technology, Ireland. He is also the chair of a steering board committee of ASM Nanocom, which he co-founded, and an associate editor for IEEE Transactions on Mobile Computing, IEEE Transactions on Molecular, Biological, and Multi-Scale Communications, IEEE Letters of the Computer Society, Nano Communication Networks elsewhere, and digital communication networks also as well. In 2018, he was the IEEE Nanotechnology Council Distinguished Lecturer. His research interests are in the area of molecular and nanocommunications, as well as Internet of Bio-Nano Things. So I will again play the pre-recorded the presentation. Start playing.
Welcome, everyone. My name is Sasitharan Balasubramaniam, and I'm currently the Director of Research at the Telecommunications Software and Systems Group, Waterford Institute of Technology, Ireland. Today, I'm going to be giving a joint presentation with Professor Josep Miquel Jornet from Electrical and Computer Engineering Department at Northeastern University, who I've been collaborating very closely on this work. And the title of the presentation is The Internet of Nano-Bio Things, Bridging the Biological and Digital Worlds. Our vision of this presentation is going to be presenting our vision for communications for 6G going forward into the future, which will be basically a revolutionary change in our view compared to the current 5G, where we will be basically merging the biological world with the digital world and physical world. What we envision in the future is that the devices that will be communicating to the infrastructure will be more widespread, and more unconventional. In this particular case, we're literally talking about biological cells. And so our vision of 6G is going to be signal conversion from electromagnetic waves to molecular signals, allowing the cyber internet to actually control miniature devices that could be within our body. We will start by presenting the Internet of Nano-Bio things, which we believe is going to be a paradigm that will be integrated into 6G. The vision that you can see right at the bottom there of the Internet of Bio-Nano things is to interconnect engineered cells, which we call as Nano-Bio things, that will have specialized communication function, and that we can actually interconnect these different engineered cells in different organs in pretty much the same way that actually interconnect devices in conventional networks. And through the connection of different molecular communication networks, can then communicate through a device called a cyber interface that will communicate to the information to the internet, effectively allowing these engineers, as I mentioned earlier, to communicate to the cyber internet. However, in this particular presentation, we only focus on one specific Nano-Bio things, and that is specifically for brain-machine interface. Many of us are very aware of the tremendous amount of technological advancement that has been made in the area of brain-machine interface. There on the example that you can see on the right-hand side is a deep brain stimulation device using a large electrode inserted into the brain, where electrical signals are actually transmitted into them to actually suppress neuron signaling, in particular for Parkinson's disease. We're starting to see new emerging applications, such as targeted neuroplasticity training, where information can be transmitted into the brain, providing new skills, which is a grand vision of the future. Unfortunately, there are a number of shortcomings with the current solutions. First and foremost, a lot of these devices are actually quite large. Even if you actually miniaturize them, the key question to ask is that how many neurons do they actually interact with? And do they actually interact with natural neurons? So what if we can actually have a combination of engineered neurons and engineered miniature devices combined together? What are the new emerging applications that can result from that? And hence, that's the reason of the Internet of Nano-Bio things. So there's a number of enabling technologies that we need to bring together to realize this grand vision. First of all, bringing in new types of concepts and theories from nanotechnologies, where we know from the field that it allows us to actually engineer to control matter at the atomic and molecular scale, and enabling communication between those devices, as well as bringing the biological aspects, in this case, synthetic biology, that will allow us to have tools to engineer circuits within biological cells. So our vision is actually presented here, and this is a joint work with Josep itself, where we want to have these engineered physical devices that you can actually see inserted into the brain, communicating with engineered neurons, so that you can actually have communication from external devices to these miniature devices, and they will actually then stimulate these neurons that will have specialized function. And this, of course, will be connected to a computing device externally, providing various different types of command, as well as reading feedback information from within the brain as well, in order to actually take corrective actions. So we're going to actually focus on two elements here in this presentation, that is at the bottom left-hand corner, the engineered cells, as well as those physical devices, miniature devices to control them. I'm going to start by giving a very, very quick background of the emerging field of molecular communications. So the aim here is basically to use communication theory and as well engineering and networking that is conventionally used in typical communication systems, but in this case, we want to actually use them in biological cells to not only characterize and understand how their communication performance is actually being performed during the communication process naturally, but also how we can maybe engineer and program them and then to have specialized functions of communications in the very, very same way that you might actually engineer communication devices through their software. So when we look at the way neurons to neuron communicate in the bottom left-hand corner here, you actually have molecules that actually transmit between the actual neurons itself. So you can assume that one neuron transmitting the molecule is the transmitter, the receiving neuron is actually the receiver, and the channel is the physical location that allows these neurotransmitters to actually flow between them. And not only can you have neuron to neuron, you can also have tripartite communications, where you have other types of glial cells that are used to maintain the well-being of neurons, such as astrocytes, that also play a role in maintaining this communication channel of neurotransmitters transmitting between them as well. So what are some functions that we can actually gather from molecular communications? And what can we actually do with them? So one thing that we're actually very, very keen in doing is to look at new forms of molecular computing functions that can use communication. So in the very same way that you can see on this slide that you have digital logic gates that are built out of silicon technology, which most of us are pretty much aware of. Here are examples of an OR gate and AND gate. Our question is that, can we actually take this very same design that is used in silicon technology or other types of physical technologies and apply them to biological cells, and in particular, cells that are actually within the brain itself? So we started to look at how we might be able to, let's say, engineer neurons as logic gates. And you can actually see that on the bottom left-hand corner here as an example, where we might be able to combine 3 different types of neurons together, whereby 2 neurons would actually act as an input, emitting signals, it could be neurotransmitters, that goes into the 3rd neuron that will then interpret that, and based on the engineered threshold, will then basically emit a certain amount of output that is coming out there. So you can actually see on the right-hand corner here, the very top graph here, an example of an input. The very second graph there is the other input, and the 3rd graph right at the bottom there, the 3rd row, is basically the output cell. So you can see here in the example here, in the 2nd column, if you actually have a 1 input going in with no signal in the 2nd input, you can still get a 1 output coming out. And this is basically engineering the output neuron to represent an OR gate. And in the very same way, you can also have an AND gate behavior, where you basically need 2 action potential aligned together within the same specific time slot, so that the 3rd neuron will interpret that and actually emit a signal. So what we've actually been doing is been using the neuron simulator that has been developed at Yale to see how we can, which realistically does a lot of simulations of electrochemical signaling that goes into these neurons. And there's a library of various different types of neurons out there. So what we've been doing is we've been connecting various different types of neurons together, OR gates and AND gates mainly. And you can see some example combinations on the far right-hand corner there. In the interest of time, I'm not actually going to go through the details of the specific neurons we use, but we use the various different types of neurons. And we wanted to analyze the accuracy when we're actually transmitting pulse signals into each of those input neurons to see how this will actually affect the output neuron in giving us the accurate answer for that logic operation. And you can actually see the results right at the bottom there, where, for example, in the AND gate, we actually have very, very close performance between the different configurations and types of neurons we use for the actual AND gate, compared to, say, for example, the OR gate, which is actually on the right-hand side, where we actually have more variations. And the main reason for this is because of the actual excessive noise that actually occurs between neuron-to-neuron communication. So in the case of AND gate, you actually have a much higher threshold, where you will only really get a 1 output if you actually have 2 neurons performing an actual potential, emitting a pulse into the actual 3rd neuron. So even if you actually have miniature amount of noise within the actual neurons itself, that's not going to be enough to actually activate an action potential on the other 3rd neuron for a spike to actually occur in the output, which is not the case in the case of an OR gate, which has a lower threshold. And that's why you actually see variations there. So, and also the other thing that's actually very interesting is that this also depends on the rates of spikes that is coming into the actual 2 input neurons as well. So the lower the spike rate, of course, the accuracy is much more higher compared to much more higher rate. So these information is actually very, very important when we're actually designing, finding the appropriate neurons, and also the locations that we want to put them in, depending on the rates of signals that actually goes through the microcolumns as part of the cortex. So what we've actually been doing is also looking at certain types of applications that we can actually focus on. So one of the applications we've been looking at is, can we actually interconnect these different types of neuron gates to actually do filtering? So one example of this could be for epilepsy patients, where we know from epilepsy that patients suffer from overstimulations of neurons that fire unexpectedly and fire at a very, very high rate. So what we were planning to do is, using mechanisms of actually taking a number of different logic gates, hook them all up together to see whether we can get filtering behavior, and in particular filtering at certain frequency bands. So what we've done is that we've been looking at computational models, taking the original Hodgkin-Huxley model, which is a model that is used to represent a particular compartment of a neuron, and we actually linearized that model itself. And by linearizing it, which means that we are also focusing on specific frequency bands, manipulating them, then we found out that we can actually use that to filter certain frequency signals. So what we did here, you can see, is some results from a analytical model and simulation model. You can see on the right-hand side here, we actually tried to look at different models of circuits, circuit A, B, and C. Circuit C in this particular case is all AND gates, while as the other circuits has a mixture of OR gates as well as AND gates. And we actually wanted to see how efficient they are by measuring their counter efficiency, comparing, for example, how a neuronal filter would actually compare to, say, for a typical bandpass filter that you have there. And you can see the results on the bottom right-hand corner there. And you can see here that the most effective out of all is actually the circuit C, and that each of these different circuits actually works differently depending on the frequency band you want to use, which means that we can use these different combination of neurons to create different gates and circuits depending on the application and severity of the epilepsy for the particular patient as well. So by understanding, let's say, for example, fMRI analysis of a patient during the epileptic seizures, we might be able to analyze their rate, the sparseness, as well as the location and largeness of the area within the brain that actually starts and triggers these epileptic seizures. And from that, we will be able to engineer these cells to actually insert them into the brain. They will basically connect to these natural neurons and then do the actual filtering process. So you can see here that, say, for example, circuit C, which is composed of only all the AND gates, the most effective of that is working at the cutoff frequency of approximately 60 Hertz. Whereas circuit A, you can see that it actually is more effective in much more higher frequency at approximately 120 Hertz. So what we've also been doing besides theoretical work is that we've also been looking at some wet lab work as well. And this is a work that we did in collaboration with Tampere University in Finland, where they've actually been engineered a particular type of brain cell known as astrocytes. By engineering that, we actually placed inside the actual cell a particular type of DNA. We call them engineered plasmids. And with that, we also applied another type of, two different types of solutions, okay? Those solutions are MDL29951 and T0510. And that actually invokes the calcium signaling. So working in combination with the engineered plasmid, you can then actually define specific thresholds you want before the actual calcium actually gets stimulated. So let's focus here on the fourth group of columns here. And in particular, the blue bar, which is the AND gate, and that is only the MDL solution, compared to the orange bar, which is only the T0510 solution. So you can see here that the MDL allows us to actually have an AND gate, because in order to actually hit a threshold for the cell to actually produce molecules, you need two input ones coming in. If you only had one input within each of the input ones, then you will never actually hit the threshold for the cell to actually be invoked to produce molecules. At the very same time, if you actually look at the orange bar, which represents the OR gate, you only need one input signal going in either one of the two inputs before the cell actually gets activated, right? And on the fifth column there, you actually get much more closer relationships between the two solutions that has been applied to the neuron culture, in particular looking here at approximately 50 micromoles here. So that means that, number one, our objective here is to differentiate the neuron cells so that you can have varying quantity of activation threshold for us to hit the upper limit output threshold, which we can then use that to define whether it's an OR gate or an AND gate. That's number one. And that is in particular for the fourth group of columns here for 25 micromoles. In the case of the 50 micromoles, the variation is much more closer, and you can actually apply this to neurons that actually have much lower rate of stimulation and production of calcium signals. I should also mention here is that the input molecule we're focusing for this particular types of cells are actually calcium signals that are naturally produced by cells. They emit this into the extracellular matrix, and these neurons or cells actually take in these calcium signals, and they then actually stimulate their own calcium signals as outputs. So you can see that some wet lab experiments here on the bottom right-hand corner in the center of the cell of the slide there, two examples of the AND gate and OR gate, and in particular the 25 micromole and the 50 micromole, depending on the input, whether it's a 1001 or even 2 ones going in, you can actually see that the cells are actually getting stimulated compared to very low quantity of solutions. So we believe that in the future, we might be able to engineer encapsulating devices with these neurons that house also these chemicals within them as well, depending on whether we want to create an AND gate or an OR gate, and you can actually hook them all up into miniature digital logic circuits as well. Now, if you have the actual engineered cells inserted into the brain, you're only countering the bio part of the nano-bio things. At the very same time, you may remember from my slide number 3 that you still need to have physical devices inserted into the brain to stimulate some of these cells, whether they're logic operations or even sensing them as well. So with Josep here, we've been collaborating and looking at how we can design such type of miniature device and actually using light as a mechanism to actually stimulate these neurons. And that brings us to the field of optogenetics, which Josep is going to elaborate more later in the latter part of this presentation. And the aim of optogenetics is basically to stimulate engineered neurons that will be sensitive to light at a specific wavelength. So what we've been doing with Josep has been looking at analysis of how light source from such very small nano antennas, which he's going to go through later, when they're actually emitted through the cells, how they will actually get refracted, how they will actually get absorbed or scattered, and what influence that will actually have on the transmittance of the light as they're actually penetrating through the actual cells itself. In particular, if you actually line up all the different cells. And the application for this could be being able to target specific cells for stimulation, which means that you possibly theoretically can go down to the single neuron level, or even enabling different devices, physical devices, to communicate between each other using light. So we did a lot of these modeling of these light behavior using geometric analysis. You can see at the bottom center of the slide there. And you can see that when you actually have 1 or 2 neurons, the behavior on the bottom right-hand side, the transmittance level really actually changes due to this refraction process. And what we found out is that you actually have a focusing effect of this light. And this was actually part of the experiments that we did with Josep, as well as Michael Stakiewicz, who's from the School of Medicine in SUNY Buffalo. And you can see the focusing effect here in this slide. And when you look at figure B, you see those nucleoli components within the actual cells, those black dots. And then from approximately 5 micrometers away from them, you can see no effect in slide C. But when you go further down, slide D, 20 micrometers below those cells, you see those really bright lights, showing this focusing effect that can actually happen, right? So this is good and bad. The good part of it is that you don't scatter the light all over the place to actually stimulate various different types of neurons. You can keep them focused on specific neurons. But then at the very same time, the focusing effect will also create centered heating that could actually damage the actual neuron, which means that you also have an upper bound in terms of the actual communication rate you actually want to actually stimulate these neurons. So the next part of the presentation, I'm going to pass on to Josep itself, who's going to talk more about optogenetics and other physical devices that is used to stimulate these neurons.
Okay, thank you very much, Sasi, and I'll take it away for the next few slides. So, excellent. Again, my name is Josep Miquel Jornet. I used to be at the University at Buffalo. That's how I know many of you. For the last year, I've been at Northeastern University, and throughout this period, and actually for close to 10 years, we've been working with Sasi on many of the results that you're seeing today. So at this point, we have seen that excellent. There are ways to interact with cells. We can program cells to do things that we like, And we have seen that, light can be the vehicle for the interface between the biomolecular scale and the electronic digital world. The last slide that Sasi showed us, we saw that indeed in a laboratory setup, with a full-fledged lasing system and a full-fledged microscope, we can control, we can see, we can study how light goes through neurons. And of course, that's the door to control. The reality is that, well, we're not going to embed a microscope and a full-fledged laser in anyone's head. So how can we make these devices smaller to fit the vision that Sasi showed 2 slides ago? Well, the good news is that, okay, today, thanks to the tools that nanotechnologies provide to us, we can create extremely small antennas. And as you may remember, the smaller your antenna is, the higher the frequency. And of course, if you make it very small, that frequency is going to lead you to optical signals. Does it mean that, I can do anything that I've done at radio frequencies now at optical frequencies? Well, actually, to some extent, yes. You will have to revisit a little bit the antenna theory, but at the end of the day, we can manipulate light in the same way that we manipulate the 2.4 gigahertz in your Wi-Fi, and we're able to beamform, beam steer, reflect it smartly if that's what you want. We can do all those things with miniature nanostructures and at optical frequencies. We have been working on this for several years. We did the theory, and of course, we also did some experiments in collaboration. The next step is, of course, the antenna. What is an antenna? An antenna is a device that takes a signal at a given frequency and radiates it out at the same frequency. Okay, well, the antenna is therefore just one element. I need something that can take the energy, let's say from my battery, my electrons in my battery, and convert it into a signal at optical frequencies. That device, it's a laser. And in our application, what we need is a nano laser. The reality is that for several years now, there have been several works showing how you can build super small lasers. I'm talking about lasers whose total size is just a few micrometers, and which can effectively convert your currents into photons, which is what we need. Those lasers are not just devices that will emit the signals. We can also use them for modulation purposes too. And in addition of controlling the wavelength or the frequency at which they radiate, or controlling whether they are on or off, we can also manipulate their phase, and we can even engineer them to generate fancy modulations like orbital angular momentum. And for example, in these 3 images here, this is the result of a collaboration with Professor Liang Feng, who also used to be at Buffalo, now he's at UPenn, in which we experimentally demonstrated that we can indeed generate controllable orbital angular momentum modes from a laser whose size is less than 10 micrometers. Well, that's the type of laser that we think we want to use in conjunction with our antennas. And of course, as you may imagine, if we intend to do some sort of spatial control, we need more than just one laser and an antenna. We need an array. And again, once you understand the physics of optical signals, and once you tailor the antenna theory to the peculiarities of optical signals, and in particular materials at optical frequencies, we can start talking about beamforming too. And you can either make an array that it's only an array of antennas, that's what you see on the right, but you can also make an array which is an array of lasers themselves. So for those who work on more traditional frequencies, I would say, we can do, fully analog beamforming antenna arrays, or we can make fully digital beamforming antenna arrays at optical frequencies and with footprints, as you can see here, of just a few tens of micrometers. Fine. So the technology is there. And in reality, that's the reason why many things have changed in the last 10 years. Why do I throw in this reference to last 10 years? Well, the very first time that we used the term Internet of Nano Things was in a visionary paper back in 2010 with Professor Akildis. Then in 2015, Sasi added its flavor with the Internet of Bio Nano Things. And well, we are at a point today that some of these things can become reality. And what we're going to show next are the results of some experiments in which we went ahead and we interfaced the 2 parts of this talk. We actually showed how we can use optical signals at the nanoscale to control the propagation of molecular signals. We'll talk about mostly 3 experiments which can be grouped in 2 areas. We'll start with optogenetics, which is what we have been discussing so far, and we'll then transition to optogenomics. Okay, so optogenetics. Sasi already explained it a little bit, but let's just go a little bit more in detail. What is optogenetics? Optogenetics is, in optogenetics, the idea is the following. You can take cells, and in particular in our case, because we're dealing with brain-machine interfaces, we start from neurons. We can add this specific sequence or molecule or protein, if you want to call it, which is called a channel rhodopsin, in such a way that when we shine this channel rhodopsin, or at the end of the day, when we shine a neuron with light, this channel rhodopsin will trigger the flow of calcium ions. Calcium ions are the molecules that neurons utilize to communicate between them, right? So you all know that we have neurons, and neurons form themselves into networks. The signals that go through these neurons are calcium ions. Fine. With channel rhodopsin, we can control whether these signals can propagate through a neuron or not. And with light, we can control channel rhodopsin. So with light, we can control signal propagation. That's the experiment that was very first done by, by who created the field of optogenetics. In our case, the first step was to first, let's make sure that we can reproduce the results in the literature, but then let's go beyond this. In a traditional laboratory setup, the focus is to manipulate all the neurons, all the cells in the same way at the same time. But in our vision, what we want to do is to be able to show that you can manipulate one cell at a time. Maybe the only cells that are not working in the way that you expected, or the only cells that you need them to do something, are confined in a very small region. Just to give you an idea, the size of a neuron body, of a neuron nucleus or the neuron head, it's again tens of micrometers. So if you really want to target just one neuron at a time, you need to make a device that can potentially shine light in a very small surface. So what we did in this very first experiment, once again, is we take neurons, we add channel rhodopsin to them, and then we utilize blue light to trigger channel rhodopsin, and then we utilize a fluorescent red particle so that when there are calcium ions flowing through, this particle will lighten up, and we will know that indeed there is signal propagation. What do we use to do this experiment? Well, the first step was to take a conventional laser, cap and remove the cap, block it out, so suddenly our laser is blacked out, it cannot emit any light, but then we come and we do two tiny openings, one which is just 5 by 5 micrometers and one that is just 20 by 20 micrometers. That's what you see in the figures here on the bottom, that's the openings, and on the bottom right, you see the actual light source seen with a microscope. Fine. One thing, as you can imagine, electronics and liquid media do not go very well together. So of course we had to build an encasing for this device, and as well as a holder for the dish where our living neurons were. So there was some work to do from the mechanical perspective too, but we put all those things together. And then what did we do? Well, we did the following. We took our neurons, we placed the laser immediately under the dish, a transparent dish. And then what we did was to activate the blue light lasers and then monitor the red fluorescent particle. The red fluorescent particle is the one that if there is red light, it means that there are calcium ions. And what were we able to see? Well, we were able to see that under or on top of the location where our laser was being activated, those cells would start talking to each other. That's why you see these red bright spots. Red, it's an indicator that there is calcium ions flowing. Why are there calcium ions flowing? Because we trigger them with blue light. It's okay, it's a combination of signals, but that's what it is. And what we can see is that we shine the light for 5 seconds, and then we just kept looking. We see that when we shine the light, the signals go out. But then, of course, if we stop shining the light, the neurons don't have anything else to say, so the signals eventually die. Fine. This is a result. Again, the concept of optogenetics is not new, has existed for close to 20 years. What we were demonstrating here for the first time is that instead of exciting all the neurons or none of the neurons, we can selectively trigger specific neurons. Fine. There is one thing, however, which is the following. with this channel rhodopsin, we are kind of letting calcium ions flow or not flow. Fine. Is that the only thing we are doing? Are we triggering any cellular process besides letting calcium ion go or not? And I always give the same example. When this channel rhodopsin acts in the following way. Imagine that the neuron is an elevator. Fine. The channel rhodopsin, it's nothing but someone trying to push the doors open from the elevator so calcium ions can flow. So that's a stressful process for the cell. And in other words, if you keep doing that to your elevator, if instead of pressing the button to say open the door, you have someone mechanically pushing the doors, well, eventually you will break the door, right? So our collaborator, Michal, was like, I'm quite confident we are triggering other stress processes in the cell. Why don't we go and check? So we did that. What we did was, okay, instead of just looking only at whether there are calcium ions flowing between cells, let's go and monitor two specific genes. One is CFOS, which is a gene that it's when it's triggered, it's expressed very quickly. The other is FGFR1, which is a gene that will take some time to express, but when it expresses, it controls many other things. We'll talk about FGFR1 in a couple of slides. So what we did here is to take, instead just of a single Petri dish, we take an 8-chamber slide or dish, if you want to think it, and some of the wells were with cells that had been transfected with channel rhodopsin, whereas some of the wells were just cells that were not transfected with any light-sensitive protein, just with a, with a negative control protein. Fine. What did we do then? We went again with our blue light and start pumping the neurons. Sure, the calcium ion was going around, but we were interested in seeing if there was any gene expression. And what did we obtain? Well, we indeed saw, and that's what you see in these figures, that let's look first at cells that have been transfected with the channel rhodopsin, which means that we have added the channel rhodopsin to them. And let's look at the expression of this very immediate action gene, the CFOS. And what we can see that if there is no light, the CFOS is not expressed, but when we shine with light, again, blue light, we see this CFOS gene expressed. Okay, so with light, we are controlling the expression of a specific gene. And once again, if instead of adding channel rhodopsin, we use any other not light sensitive sequence, what happens is that you can shine as much as you want, that there is not going to be any significant thing being expressed in the neurons. Fine. And this is true both for CFOS as well as for FGFR1, the two genes. Fine. What are the implications of this? Well, at this point, what we're saying is that we can utilize light to control the expression of genes. What does this all mean? Well, just remember for a second, every single cell in your body has the same source code. Source code in this case means the same DNA. And a living cell has an operating system which is executing part of this DNA. Fine. Of course, different cells in your body have different functions, which means that they execute different of the lines in the source code that you have. All the cells have all the source code, but different cells at some point have decided to become neurons and therefore only run the functions related to neuron, which are all contained in your DNA. What is very interesting is that many of the diseases, like neurodevelopmental diseases, but also neurodegenerative diseases, are all related to neurons not acting in the way they are supposed to, and in particular, not expressing certain proteins which might control communication and other cell functions. So what we're trying to say here is that now we may have a way to control or reestablish, trigger the execution of some functions which are programmed in your cells, but they were just not executed because of a neurodevelopmental problem, so they never were executed, or neurodegenerative problem, which means that at some point your neurons stop executing. Now we can say that wirelessly with light, we can trigger the execution of those functions. And this whole thing, it's at the core of this paper that we published last year, again, in collaboration with Michal, Liang, and many others, in which we were actually talking exactly about how you bridge biological networks and the electronic digital world in the context of brain-machine interfaces, at least. And we draw this parallelism between how in a computer you have your operating system, you have your applications, you have inputs and outputs to your applications, and then things happen. Well, in a neuron or in a cell, the thing is the same. Now your source code is your DNA. Part of that source code is run by the operating system, but then you can have inputs and outputs to trigger different applications that are already installed in your source code. So what we're saying here is that light can be one of those inputs. Of course, we talk about trying to do something good, which is to reestablish functions that at some point were lost. The media always likes to take spins of things, and they claim that, you know, some people are hacking the genome and blah, blah, blah. Sure, I imagine that you can, you know, sell the technology in different ways. Okay, so what we have seen so far is that with this channel rhodopsin and blue light, what happens is that we know we trigger some genes, but is there a way I can control only one specific gene? And if yes, what is the gene that I should be targeting for different applications, for example, in our experiments here? Well, to answer the first question is how can we increase the specificity so we only target one gene at a time? Well, there are many ways to do that. We propose to go with a molecular toggle switch. What is that? Well, let's look at this figure on the right, and let's start with A at the top. There are some molecules that they are attracted when they are illuminated with light. And in particular here, we use two proteins, FIB and PIF6, in such a way that when we shine light at 650 nanometer, that's kind of reddish, red light, the two proteins will bind together, the two molecules will bind together. Fine. What's interesting is that if you attach things to the tails of these two molecules, when the molecules are attached, those extra proteins, molecules, sequences, whatever you call it, attached to the tail of these molecules will also be in contact. So you can use light to bring any type of protein that needs to be expressed, of molecule that needs to trigger something, you can bring it together. And not only that, then we can come with light at 750 nanometers that will break this bond and detach the molecules, so we can switch them on together or off apart. So we can use light to control the expression of individual genes by utilizing this toggle switch. Okay, which gene should we target? Well, in this case, again, this is a collaborative work with professors Michal Stachowiak and Eva Stachowiak, both at the University at Buffalo. And what they have been working for several decades now is that there are different types of genes. Of course, some genes control some very specific functionalities. Some genes are known as master genes, genes that are in charge of many processes. And one of those master genes is FGFR1. In particular, this gene is in charge of triggering when a cell needs to evolve from being a stem cell into becoming a specific type of cell. And in particular, in our experiments, we're going to be working in how we can control FGFR1, and in particular, its nuclear version, so N-FGFR1, to make a stem cell go to a full-grown neuron, or, important, to go from a full-grown neuron to something that resembles a stem cell. What did we do? Well, the same type of experiments, but now there is no channel rhodopsin anymore. Now there is this molecular toggle switch. And again, same type of light. In this case, it's not blue, it's red and even more red. And there are some time scales. Remember, FGFR1, it's a gene that takes its time to express. So we are talking not about seconds or minutes, we're talking about hours. The outcome, well, the outcome is what you see in this figure. The top and bottom figures on the left that you can see here is the result of having neurons transfected with the molecular toggle switch attached to FGFR1. And what you see here is that if you don't shine light, FGFR1 now it's not being expressed because that molecular toggle switch is off. But when we shine light, you see how things at first you couldn't tell what they are, which are, kind of randomly shaped things that will eventually be neurons. Well, when you shine light, you can see how their shape changes and they start becoming full-fledged neurons. And as you may know, the first thing that neurons do when they are happily growing is to create connections to other networks, connections to other neurons. So eventually they can create something that resembles the human cortex. So we have been able to show that with light, we can go from something close to a stem cell to something that starts to look like a neuron. And if we break this process, the neuron will actually go back to something that's looked like stem cell. Fine. At this point, we are done. We're done for today. What we have been doing with Sasi is to share what we think is the vision for the future of communications. At some point, many people think that this is science fiction. We actually want to think that this is science. And actually, when we say science, it doesn't mean a place for scientists to work. It also means a place for engineers to work, because we need to make this physics into reality. We have shown you today how we can use the digital wall, a computer, to control in real time the biological wall, a bunch of human living neurons. There are many still challenges to realize the vision that Sasi showed at the very beginning.
Many people think that this is science fiction. We actually want to think that this is science. And actually, when we say science, it doesn't mean a place for scientists.
It's an opportunity for many of you who are here, and I hope that moving forward and talking about wireless communication,
we have shown you today how we can use
you don't forget about
the digital wall, a computer, to control in real time the biological wall, a bunch of human living neurons. There are many still challenges to realize the vision that Sasi showed at the very beginning of how do we think that brain-machine interfaces will look like, but that vision is not science fiction anymore. It's it's an opportunity for many of you who are here, and I hope that moving forward and talking about wireless communications, the Internet of Things, you don't forget about this part and join us in this quest. The ultimate connectivity.
What do you know?
Yes. And thank you very much. And we're now open for questions.
Okay. The very end of the video, there's something wrong. There's another source of the audio. But again, thanks again to our two speakers. And so I guess now we can take some questions. Dr. Jonathan is here. Very nice to see you again.
Yes, again, I'm happy to see. Actually, I was going through the list of participants, so many people and so many known names that, it's exciting. And I apologize for going over time on a Friday afternoon, but okay, we tried our best to condense all this information in one talk, and we want to thank you for giving us the opportunity to share this work. That it's with Sasi, it's with Michal, it's with Yongho, it's with Liang, many people at UB, and I know that some of you think it's crazy. I just think that it's a matter of time. I actually see some comments on the Q&A. Maybe I can read one of the questions, which is, how many years will it take to build functional and reliable sensors for any of these applications? Well, we are building functional sensors for this, and I think that hopefully with the experiments you have seen that this is no longer just a paper to write on info theory or communications theory and to send to some transaction journal. This is a type of paper that today it's more about, okay, this works, we can show it. Now what we need is more people to help us make it practical. So again, it's not science fiction. It's more science and engineering.
Thank you. And is there any other questions coming from the audience? While waiting for the new question, can I ask you one question?
Yeah, of course.
You mentioned the science and the information theory, communication theory. Do you think that this is possible that you generate new theory? Because anyhow, we do now know what is the the information, again, structure, and also the communication, the procedure, also the signal structure, right? How the information is carried in the neural, in the DNA. Do you currently it seems that if you attack that from like a black box, again, the strategy is more like a engineering problem. But do you think it is also possible to generate a science that will fundamentally model this process and also give us theory about this whole process?
That's a very good question. What I would say is the following. many times we have people talking about, a brain-inspired computing and machine learning and artificial intelligence when we don't yet know how real intelligence works, right? Or how really the brain works. So I think of this platform as two ways. On the one hand, we can use the technologies we are developing to study and understand what type of signaling, what is going on. So, for example, when we work with our colleagues in the medical school, Michal, Eva, Yongho, when we work with them, right, I want to think that the communications is the final application, but for them, I'm just a tool that will allow them to study what's really going on. So it depends on how you sell the story. Do we need a new theory? Yes, in the US, there is the Brain Initiative. In Europe, there is something similar. So it's all about studying the brain. Then we can start talking about how to interface with it and not. Right now, we're making interfaces that we know that are doing things. We still can learn a lot. And with these interfaces, we can learn too. I would say that there have been many papers on, modeling signal propagation between neurons. We actually have an interesting paper coming out soon, again, with the same folks, Michal, Yongho, Eva, in which we actually follow a machine learning approach to understand what is being triggered. And again, many people these days, it's easy to throw machine learning in the loop, just your paper looks fancy. In our case, think in the following way. We're talking about thousands of genes that are expressed, so we need to study the relations of why are those genes being expressed. And in that case, you can have one student for 10 years trying to look at connections, or you can throw in a machine learning algorithm. So we did some of these results, and we are now in the second round of review, so hopefully that paper is up soon. But so the answer, we need a new theory, yes. We don't know how things really work, that's true. And we think that this platform can help you not just to execute, but also to study and learn.
Great. Thanks a lot. So let's just take the last question because the time is very limited. So here's one question from the audience. Look, how can we make sure that we are targeting the wanted neuron? How selective is the process?
Very good question. So at this point, the only thing that we can control is the spatial control. So usually when we talk to the bio community or the medical community or the neuroscience community, they have not been too worried about targeting one neuron or another. They have been worried only about targeting one gene. Fine. What we're bringing here is targeting one gene in one neuron. And right now that's accomplished through literally a spatial selection. So spatial multiplexing, beamforming, things like that. Of course, you will say, sure, how are you going to do beamforming in the brain? Well, those are many other challenges. Light allows us, remember, why do we go with optical signals? For many reasons. One of them is that the higher the frequency, the smaller the wavelength. Everything you know about antenna theory applies here too. Therefore, you can do beamforming and have control resolution in the order of the wavelength. So, a few hundreds of nanometers, I would say few micrometers makes sense. But right now, the control is literally through space. Of course, usually a follow-up question that I get here is that, sure, and how are you going to put this in someone's brain? Well, that's one of the challenges right now. But for example, some of you may be familiar with the DARPA program from 2 years ago about neuron nanotechnology, NQ. Which the entire point of the program was exactly to look into this, ways to augment function of the brain through nanodevices that might be injected not through the skull, but actually through your nose. So in the same way that you can sniff some things, you could also sniff some of these devices. And again, this is not Josep Jornet, this is DARPA in an official program solicitation. So I guess that maybe we're onto something, but these are all very good questions, and that's why we need more people to join this community. And I think that the comms people have an opportunity also to, again, connect the ultimate machine to the internet, not just your temperature sensor, but your brain.
Fantastic. All right, so thank you so much again, Josep.